Hello everybody. All right, so today we're going to do day 3 of mitosis and then finish that off and move on to just a one-time kind of topic here. All right, let's start off by calculating a mitotic index for this picture of onion root cells. I know they're onion root cells because they're all square, and scientists love using onion root cells. So let's make a little table for ourselves. We have interphase, prophase, metaphase, anaphase, and telophase, and we're going to be looking for how many of each there are. So you can choose to do that while pausing the video and then checking back to see what I put down, or you can kind of follow along with me. Special note here, like there are a couple of weird ones on this one, like right down here. I mean, there's not even a nucleus that we see there, so we're not going to count that one at all. And then right over here, there's this like weird area where it's like a couple of cells like sitting on top of one another. And it's hard to tell what's going on there, so we're also going to ignore some of those things there. All right. Right now, you can either pause the video and do this one on your own, and then check back with us, or you can follow along with me. You know what I'm going to do is I am going to add that picture of the different phases in here. Here we go. I'm going to move this guy down here to kind of help you see what is what. All right, here we go. I'm going to start up here in the upper right. This is definitely an interphase because we cannot see any stringy chromosomes there. Interphase, interphase, interphase. Ooh, this one right here might be prophase because we can see some white space in there, so it's definitely a little bit stringy. Oh, this is a great telophase right here because we can see the cell wall not completely fully formed and the chromosomes are just kind of unwinding here, so that looks like a telophase. Here's a good prophase. We're not counting this one, we're not counting this one. Here's an interphase and an interphase and an interphase. I'm going to ignore, I'm going to ignore these other ones over here. Here's a prophase because we can start to see some white space in there. I'm ignoring all of these here. Inter. There's a good prophase one right here. Inter. Prophase. Can't quite tell this one. And I am going to ignore all of these because they're too faint for us to really make a solid description of. And these I can't even see a nucleus in. Inter. And don't be surprised that you see so many in interphase. Here's a couple of prophases here. Ooh, finally we have a metaphase. This one's lined up nice and easy right along the middle here. Yeah, don't be surprised when you see a lot of interphases, because remember, cells are not dividing all the time, and because they're not dividing all of the time, you're not going to see a lot of cells in mitosis. This one's an interphase because I can see this dark nucleolus right here. Oh, here's a good anaphase. Look, like you can see the cells pulling apart from one another and no cell wall in between. These are all kind of ghosty here, so I'm ignoring those. Here's another anaphase. There's a couple of prophases. Inter. Really good prophase. You can see the stringy chromosomes there. I'm going to assume that one is as well. Inter. Prophase. Another anaphase. The rest of these are all interphases because we don't see those stringy chromosomes at all. Whoops. All right. Okay, I would pause the video. I'm going to pause my video right now just to count all these up. Why don't you do the same? Okay. Here's what I came up with. If we're off by one or two from each other because I missed one or you added one, it's okay. It's not a big deal. A couple of these are real subjective sort of ones, and so it would be, even if this was like a quiz, it would be easy for you to be just a little off from what I have, and it wouldn't make either of us wrong. There's some clear ones. For example, if you were to mark this one here as telophase or anaphase, that would not be correct. This is a much better metaphase because those chromosomes are lined up right along the middle, and you can see kind of the strings pointing out there. Okay, so we have 18 18 cells that are in mitosis. We have 66 total cells. So to find our mitotic index, we take 18 divided by 66, and we get 0.27. So this is a little bit like a percent, but we don't turn it into a percent. So we say that about 27% of these cells are in mitosis. And that's where we are. All right. Let's talk today about... I'll draw a little wall. Let's talk today about DNA replication. And when we talked about mitosis, we mentioned that part of interphase is the S phase, and that is where the DNA replicates or copies itself. And this is really important because we have a 2N cell. And after S phase, we have a cell that has 4 of each chromosome. 2 of them are exact copies of each other. And then in the process of mitosis, that 4N cell is going to divide into 2N. Oh, I made that one so small. And the key here is so that our daughter cells are genetic copies of our parent cells. And that cannot happen without this S phase doubling of all of our DNA here. And so today we're going to talk about how that happens. Now there's an activity for you to do that goes along with these notes. And there's a lot of different ways we could do this. You could be listening to the notes and going through the activity kind of at the same time to get an understanding about how this works. I could have you do the activity first to get some learning and some of the ideas under your belt so that the notes strengthen what you learned in the activity. Or you could do the notes first and then go do the activity so that the activity puts to work what we talk about in the notes. Right now is your time where you're going to make a decision about what you want to do. Are you the kind of person that likes to do the stuff hands-on first and then get the notes for it, or do you want the other way around? So the directions for how to get the activity are in the remind, but I'm going to move ahead now with the notes. So you could either pause the notes right now and go do the activity, or you can do these notes right now and then go do the activity. All right, let's talk about DNA replication. So we've got our double helix here. We got our double helix, that is our DNA, and we know that there's a bunch of bases that connect to each other going across here in this picture. So how do we double this up? Because this is like a zipped up zipper. How do we make sure that this doubles? Well, the answer is pretty easy. First thing that has to happen is that the zipper needs to get unzipped into its 2 sides. And then a new strand is going to be added in here that is going to use the original strand as the directions for what should be on it. And so each new DNA molecule that is made is part old DNA and part new DNA. And understanding DNA replication is really, really, really important because if we understand how it works, we can understand what happens when it goes wrong. Or we can take advantage of our knowledge of this and use it to develop maybe drugs or medicines that will maybe target this to kill things. For example, some antibiotics will prevent the DNA from replicating. And if the DNA can't replicate, then the bacteria can't reproduce. And if the bacteria can't reproduce, then it doesn't grow and cause harm or damage to your bodies. And so understanding DNA replication is really incredibly important. So this is a pretty easy process for how this works. It involves the use of two enzymes. One of those enzymes is DNA polymerase. And DNA polymerase is an enzyme that is going to kind of attach itself somewhere here on this new strand, like right here. And if it sees an A on the old strand, it's going to bring in a T on the new strand. And then it moves on. If it sees a G here, then it's going to bring in a C here. So we know it's an enzyme. We know it's an enzyme because it ends in ACE. And it builds onto the DNA polymer. So it's the thing that is going to build onto it. So a couple of vocab words here that go along with this. The original strand. So the original strand is what we call the template strand. And that's what it's going to use to make the new one. So if it sees a T, it's going to use that template to make an A. And the new strand is the one that is synthesized or made. It uses the normal complementary base pairing rules. And by using the original strand as the template strand, you can be sure that the new strand is an exact copy of the old strand. And that's really, really, really important. The other enzyme that is needed is an enzyme that needs to unzip the DNA so that the DNA polymerase can be doing its thing. Here's another DNA polymerase moving down that way. This enzyme here is called helicase. How do we know it's an enzyme? That's right, Simone Thornton. It ends in ACE. And this one's really cool because it unzips that helix. We call it helicase. So it does the unzipping of the DNA. And that allows the DNA polymerase to go in there and do this. Now, next year in HL, you're going to learn a lot more information about how DNA replicates. It's actually a really complex process. The only thing you need to know this year is that DNA polymerase adds the new bases on to make the new strand. It uses the original strand as a template and synthesizes a new strand off of that. And helicase is the enzyme that unzips this double helix to expose those strands so that the DNA polymerase can do its work. Part of what you're going to do in today's assignment is you are going to be checking out a video just to make sure that you kind of understand how this works, like visually. Now, the rest of these notes are going to be about two scientists that figured this out through like a very innovative little thought process. I don't need to write that in yellow. And their names are Meselson and Stahl. These are the two scientists that figured out that how DNA replicates the way it does. Now, they had a couple of ideas. They thought that DNA could replicate in three ways. Those three possibilities, those three ways that they thought DNA could replicate were conservative, semi-conservative, and I didn't plan out my space well enough, so I'm going to have to put the other one down here, dispersive. So, in conservative replication, we have our DNA molecule here, And we always start with a single DNA molecule here. So I'm going to draw my double helix in a bunch of spots. Okay. And the idea here is that in conservative replication, the original DNA molecule stays together. And they had no idea how this would happen, but they thought, well, this is one way that it could happen. And the new DNA molecule is a completely newly synthesized DNA molecule. That's conservative replication. And once again, this is a possibility. Messelson had no idea what it would be, but this is a possibility. In semi-conservative replication, they thought, well, maybe one strand will stay the same, and the other strand will be brand new. And then dispersive replication was the craziest one. And this one's like ridiculous to try to draw. So if you don't want to draw this one, I totally understand, because I don't want to draw this one. In dispersive replication, yeah, do you guys see this here? In dispersive replication, the idea was that each DNA strand is like part old DNA strand. I don't even know how to do this. And part new DNA strand. And so, I mean, imagine those are good double helixes, but they're not. That's dispersive replication. That idea was the craziest. So, how do they do this? Well, they did this by doing the same thing that we have talked about multiple times in the course of studying DNA. They used radioactive materials. Specifically, they use radioactive nitrogen, because remember that every DNA base has those complementary base pairs that are nitrogenous base pairs. Okay, so nitrogen can exist in two isotopes, one being a little bit heavier than the other, and those are called nitrogen 15 and nitrogen 14. So what they did is they grew a bunch of like bacterial DNA. They grew a bunch of bacterial DNA. They just let a whole bunch of bacteria grow. And they grew it in this heavy nitrogen right here, which means that we're starting with DNA that is made of all nitrogen that is nitrogen 15. All nitrogen that is nitrogen 15. Okay? And what these guys did a whole bunch is that they, what's the word I'm looking for? I lost my train of thought. Anyway, they took this bacteria that was grown in this heavy nitrogen, and they would take a bunch of it out, and they would spin it in a centrifuge. A centrifuge is that thing that you've seen on CSI that like spins blood samples around really fast and things like that. And you can do this in your house if you take a bottle of like Italian dressing and you put it in your arm, and then you need to start windmilling your arm a whole bunch. All of the heaviest stuff in the dressing will sink to the bottom, and the lighter stuff will be at the top. And since nitrogen 15 is heavier than nitrogen 14, they used some radioactivity to illuminate the different bands, the different locations about where this nitrogen was, and that would show them where the DNA was. So right now, at the start, since all of the DNA in the bacteria was of nitrogen 15, when they spun it around in the centrifuge, they ended up with a whole bunch of nitrogen gathering down near the bottom. And this was all nitrogen 15. So depending on where all of this DNA ended up, they could have an answer. Okay? So how does this work? Let's do some predictions now. Okay, so I'm going to be bouncing back and forth between my drawing at the top and down at the bottom. All right. Let's start with our conservative replication. So, in conservative replication, all of our DNA is nitrogen 15 at the start. And after copying once, half of our DNA will be the original old nitrogen 15 DNA, and the other half of the DNA will be new, lighter nitrogen 14 DNA. So, after the first replication, what we should see in a test tube is a bunch of our original nitrogen 15 DNA, and then a bunch of our new nitrogen 14 DNA. So how does this work with semi-conservative replication? So let's look up at our semi-conservative replication here. Each DNA strand is half new, half old. So after one replication, all DNA is going to be half nitrogen 14 and half nitrogen 15. So in our test tube, we should see a line in the middle because the DNA has nitrogen 14 and nitrogen 15 in it. Okay, so notice when I drew this, like I made sure to draw it in between where the nitrogen 14 and the 15 lines were. What should our dispersive model have looked like? If the dispersive model was right, what Meselson and Stahl would see, let's scroll back up here, is a really similar model to the semi-conservative replication, since each DNA strand is about half new, half old. What we should see in this case is a line right in the middle, because all of the DNA is part nitrogen 14 and part nitrogen 15. So how do you tell the difference between these two then? Since these two are really similar to each other, what you have to do is you have to let them all copy again. Because when they copy again, that's when the differences will really start to shine. So, let's think logically what's going to happen here. If the original DNA strand stays the same, and the new one is using our lighter nitrogen 14, well, what we should see is our original one staying there. But all of our new DNA being made will be nitrogen 14 DNA. So when this one here splits, it's going to be made with all of our new nitrogen 14 stuff. And since our original DNA was nitrogen 15, when this one here replicates, we will get one of the original nitrogen 15s and then another nitrogen 14. So at this point, we should have 3 times as much nitrogen 14 DNA as nitrogen 15 DNA. So what we'll see is a much thicker line here at our nitrogen 14 compared to our nitrogen 15, because we have about 3 times as much. In semi-conservative replication, we'll still see our line right in the middle, because there will still be DNA that contains the original nitrogen 14 with some nitrogen 15 DNA. But now, because our new DNA can only be made using the nitrogen 14, we're going to start to see pieces of DNA that are just made with that isotope of nitrogen 14. And then in dispersive, since all of our DNA is a mix, we're just going to see a line right here in the nitrogen 14 and nitrogen 15. Now we have some real differences here. All right, are you ready to see their actual answers? So I've got a picture here of their actual data. All right. So, here is the original nitrogen-15 set of DNA here. Now, you can see right down here we got this nice big thick black line at the nitrogen-15 line. Now that we knew that would happen. You saw that right up here. Okay, we knew that would happen. So bacteria can reproduce really quick. So after 20 minutes, they pulled out a sample of the DNA and they spun it around. And so at 20 minutes, right here, we get our first generation. And you can see that this line is right in between the 14 and 15, which means that that axes out our conservative replication, and our answer is either that it's going to be semi-conservative or dispersive. So at 38 minutes, they pulled out another sample, and look at this right here. You can see a line at the nitrogen-14 level and the nitrogen-15 level. I'm sorry, at the nitrogen-14-15 level and the nitrogen-14 level, which means that it totally matches our semi-conservative prediction. And take it even further, at the nitrogen-13 level, you still have a little bit of that mixed one, but the new DNA, which is all nitrogen-14, gets thicker and thicker and thicker as you go on. Meselson and Stahl showed that DNA replicates semi-conservatively. Now, you knew that already because of what we did right up here, but now we have a name for it. That name is semi-conservative replication, which means that each new DNA strand is part new synthesized DNA and part original template strand. And that's it. Everybody stay safe.